Hi, we are continuing our PowerPoint. This is just a review of the law of independent assortment, which is the segregation of one gene does not influence the segregation of another gene pair during meiosis. So again, this means that the alleles, they do segregate independently of each other, and again, this is during the process of meiosis. This is an example of a dihybrid cross. Before, the problems that we saw were monohybrid crosses. Monohybrid crosses are crosses that involve one trait. A dihybrid cross involves two different traits. So you see here that the P generation, we are crossing a round yellow P with a wrinkled green P. And all the F1 generation are round and yellow, but they're all heterozygotes. So when we cross to the F1s, or the two heterozygotes, we get a very different Punnett square in the F2 generation. And we do get what we call a 9:3 to 3:1 ratio. Again, this is a cross involving two different traits. Okay, so we do have a concept with lethal alleles. This would be an allele that causes death. Okay, and there are some examples with the hairless trait in dogs. Okay, and again, this is a little bit different because it does not follow Mendel's laws, because typically the homozygous recessive is typically the actual problem phenotype. Okay, this is just giving you an example of that cross. Multiple alleles occurs when there is more than two alleles in the population. So this one is giving you an example with rabbit coat color. Another example is with human blood types. We do know that A, B, and O are three different alleles for blood types that give us blood types A, B, AB, and O. So there's two different examples here for multiple alleles. Rabbit coat color, which is in the chart here, and then the blood types. And we will do a problem like that in another PowerPoint. Another thing is epistasis. This is when one gene masks the expression of another. And this can be seen with the Bombay phenotype, all right? And it also can be seen with mice coat color. All right, so just so you know that the glycoprotein that attaches A and B antigens to the surface of red blood cells. If an individual is homozygous or heterozygous, big H or big H lowercase, the glycoprotein is produced and the A and B antigens attach themselves appropriately. If the individual is homozygous recessive, or little h, the glycoprotein is defective. So A and B antigens do not attach to the red blood cells, even though the individual may have an ABO genotype, all right, indicating type AB or AB. So then that would make that person blood type O. Another type of a problem is incomplete dominance. This is observed in snapdragons, the typical flower. Now, what they saw a blending of colors. When they crossed a red with a white snapdragon, they saw pink flowers. But then, when they crossed the two pink snapdragons, they did find a ratio of 1 red to 2 pink to 1 white. And that is shown on the two different boxes that came up. Codominance is what we find when we have blood types, because they are both expressed equally. So the blood type AB, that means that A and B are both expressed the same. So that gives us the blood type AB. Okay. Oops. So when we talk about human genetics, a lot of it is a little bit more complex. And that is because of polygenic traits. Height, eye color, skin color follows along with this. And what happens is it's an additive effect of two or more genes of a single phenotype. Okay? And what you see here is you kind of add up all the capitals, and that gives you the variation of the coloring that we get. Multifactorial traits are determined by the combined effect of one or more genes plus the environment. And that is with things that we have a genetic disposition for, but the environment can help aid in that. So body weight. Some of us are born to be very thin or very large, but what we decide to do in our environment, well, that will play along with our genetics. So it does control by a lot of genes plus the environment. So that is the end of this PowerPoint. We will continue on with sex-linked genes next.